to educator.com. We're going to continue our discussion of cell structure and function by focusing on cell membranes and transport across cell membranes. We'll begin by talking about the structure of cell membranes. Cell membranes are composed of a phospholipid bilayer embedded with proteins. Right here is the chemical structure of a phospholipid. And phospholipids have a particular property of being amphipathic. Amphipathic refers to the fact that they have one region that is hydrophobic and the other that is hydrophilic. The fatty acid tails right here are hydrophobic. They're attached to a hydrophilic head region composed of glycerol bonded to a phosphate group, which may in turn be bonded to another group. Right here, the other group is choline, although it could be serine or another group. So this region is hydrophobic. The phosphate and its attachment groups are hydrophilic. Therefore, this is an amphipathic molecule. Because of this, phospholipids congregate into bilayers, and this allows them to sequester the fatty acid tails, which are hydrophobic, from the water on the outside of the cell. So this is a typical phospholipid bilayer structure with the hydrophobic region on the inside and the hydrophilic heads pointing outwards, exposed to either the cytoplasm or the extracellular space. This particular phospholipid is called phosphatidylcholine, and that's named after the type of bonds that form it, as well as this choline group out here. You could also have another phosphoglycerol, so it has the phosphate group in the glycerol, but again, it might be phosphatidylserine or another group. As I mentioned, the phospholipid bilayer contains proteins, and actually, the cell membrane can be up to half protein, and then the other half phospholipids. This description of the cell membrane is known as the fluid mosaic model. And let's look first at the word mosaic. So mosaic is referring to the fact that it's composed of a phospholipid bilayer embedded with proteins. So here this shows, as I mentioned, the hydrophilic head regions pointing out and the hydrophobic tail regions sequestered within the cell membrane. There are two major types of plasma proteins, and those are peripheral proteins. Peripheral proteins are loosely associated with either the outside of the cell, so they're loosely associated with the membrane, either at the extracellular space, so let's say that this is the extracellular side, and that this is the intracellular or cytoplasmic side. So peripheral proteins are associated with one of these sides, whereas integral proteins are embedded within the cell membrane. These proteins are also amphipathic. They also have a hydrophobic region and one or more hydrophilic regions. So as expected, the hydrophobic region of the protein is here, embedded in the area where the lipids, which are hydrophobic, are also found. And the hydrophilic regions can extend outside the cell, either into the extracellular space or in the cytoplasmic space. These proteins don't always traverse the entire membrane. They may actually just be partially embedded. So you could have a protein like this, which is sticking out into the cytoplasmic side but doesn't go all the way through. Those proteins that do go all the way through from one side to the other are called transmembrane proteins. Another important feature of cell membranes is that cholesterol is embedded among the lipid bilayer. So there will be cholesterol molecules in here, and they help the cell membrane to maintain its fluidity. So the cell membrane is not immobile. Everything doesn't just stay packed in there. There's actually movement, but the movement is lateral. So phospholipids can actually switch places. They can move back and forth, and they can actually, this one might switch places with the one adjacent. The same thing can happen over here. Rarely, a phospholipid from one layer will actually flip with one in the opposite layer. Mostly the movement is lateral. So movement within the cell membrane is mostly lateral. And this also applies to proteins. Some proteins have the ability to actually move around laterally within the lipid bilayer, although others are actually fixed in place by the cytoskeleton, by elements of the cytoskeleton. So again, this fluidity refers to the fact that these are not just stuck in place. Things can move around. Now, how does cholesterol help to maintain this fluidity? Well, a couple of ways. First of all, because these cholesterol molecules are wedged in here, they prevent the fatty acid tails from packing together. So it keeps them, instead of being packed together where they can't move, there's a little bit of space in some of these regions, and that allows for this lateral movement. In addition, cholesterol stabilizes the membrane by preventing it from solidifying at low temperatures. And the overall consistency of the cell membrane is similar to oil, like the type of oil that you'd cook with. These membrane proteins play very important functions within the cell, and we're going to talk in detail about some of these functions. But to give you an overview for right now, one function of membrane proteins is that they may be enzymes. Since they extend into the cytoplasmic space, or at least some of them do, they can actually catalyze reactions within the cell. So there may be enzymatic activity in this region. So that's one function. They can function as enzymes. Second is to allow for adhesion to neighboring cells. And there are various structures such as tight junctions and gap junctions and desmosomes that we're going to talk about that allow for communication between neighboring cells and to hold cells together. So some proteins which extend into the extracellular space actually allow the cell to adhere to neighboring cells. Third function is transport. The cell membrane is only selectively permeable. It only allows certain substances to pass through the phospholipid bilayer. So in order for other substances to pass through, they need a transport protein. So there's a couple ways in which the proteins can transport it. It can be a channel, or it can undergo a conformational change. Either way, some of these proteins act as transport proteins. And that's why, if you look at this one, it's traversing the entire cell membrane. So it could function as a transport protein. Fourth function, proteins in the cell membrane may be receptors. We're going to talk in the next lecture about cell signaling, but one major way that cell signaling occurs is through receptors. So molecules can, from another cell, for example, can come along and bind to a protein and signal the cell to grow, change in metabolism. They can be invaders that are being engulfed. So again, intramembrane proteins can act as receptors. And finally, recognition. Cell membranes have particular types and amounts of protein depending on the type of cell. So a cell in the GI tract will have different membrane proteins than a neuron. And these proteins allow other cells to recognize the cell type. And actually, what's very important in recognition are glycoproteins. So you can tell from the name that this is talking about proteins it's actually carbohydrate chains usually that are bonded to the protein. So you'll have proteins on the cell membrane that are extruding out into the surface of the cell, and they'll have these particular carbohydrate side chains that label the cell. Actually, membrane lipids can also help out with the labeling. There are also glycolipids. So carbohydrate chains on the lipids in the lipid bilayer can also help to label the cell. Okay, so now we've looked at the structure of the cell membrane. Our next step is to discuss how substances are transported across the cell membrane. Cell membranes are selectively permeable to substances, as I mentioned. What this means is that some substances can cross more easily than others. Some substances cannot cross at all on their own. This allows the cell to control what enters and what leaves. Permeability is dependent on several factors, in particular size, polarity, and charge. First, looking at size. Small molecules are more permeable to the cell membrane. Very small molecules can much more easily get through the cell membrane than large molecules. So the cell is less permeable to large molecules. Polarity. Thinking about the structure of the cell membrane, because of that hydrophobic internal region, a molecule that can cross easily is going to be a hydrophobic molecule. It's going to be a non-polar molecule. Polar molecules have a much harder time crossing, although if they're small enough, like water. Water is polar, but it's small. So a very small polar molecule can cross, but in general, the ones that cross most easily would be both small and nonpolar. Charge, again, because of that hydrophobic central region, an uncharged molecule is going to have an easier time crossing than a charged molecule, such as ions. So ions can't cross through the cell membrane on their own. They need the help of a transport protein. Glucose is a larger molecule. It's polar. It also is not going to cross the cell membrane very easily. Looking at examples of substances that do cross the cell membrane more easily. Those are things such as oxygen, which is small. Hydrocarbons, hydrophobic. I talked about water being small and yet it is polar. CO2, small and nonpolar, and urea. So these all can cross relatively easily, whereas larger polar molecules, ions like glucose, sodium, hydrogen, and calcium, all are going to have a difficult time crossing, if they can even cross at all without help. There are two general types of transport across the cell membrane. The first is passive, and the second is active. The major difference is that active transport requires energy. And in the cell, the main form of energy is ATP. So active transport requires the input of energy in the form of ATP. Passive transport does not require the input of energy. In this slide, we're looking just at passive transport. And there are two types of passive transport. The first is simple diffusion, and the second is facilitated diffusion. Starting out with simple diffusion. Simple diffusion means that the substance that is crossing the cell membrane does not require a transport protein. So first of all, neither type requires energy, and you have to consider why that is. It's actually because with diffusion of either type, a molecule is moving down its concentration gradient. So with diffusion, a molecule is moving down its concentration gradient. Substance moves down its concentration gradient. What do we mean by down its concentration gradient? Well, an area of higher concentration to an area of lower concentration. Higher to lower concentration. For example, imagine that on one side of the cell, I have a lot of oxygen. So we'll have oxygen represented as just these red dots. And let's say I have a lot of oxygen molecules over here. And as you know, these can cross the cell membrane on their own. They do not need the help of a transport protein. Over here on the other side, let's say I have a lower concentration of oxygen. Oxygen is going to want to move from the region of higher to lower oxygen concentration. So it is going to want to go in this direction. Input of energy will not be required. Now, if I wanted to move oxygen from this side to the other side, it would require the input of energy. And the molecule is only moving down its own concentration gradient. So there could be, let's say, CO2. There could be tons of CO2, putting that in blue, on this side. And that is not going to prevent the oxygen from moving in this direction, even though there's a lot of solute on this side, because it's only considering its own concentration gradient. The CO2, if it's moving down its concentration gradient, is actually going to move in the opposite direction. All right, so that's simple diffusion. In simple diffusion, we're considering substances like oxygen and molecules like CO2 that do not need the help of a transport protein. They can actually cross the cell membrane. Facilitated diffusion refers to substances that are still moving down their concentration gradient, but they require the help of a transport protein. For example, glucose. Glucose is not able to cross the cell membrane on its own generally, and I may have a lot of glucose on this side. So yellow is going to be glucose. So glucose naturally wants to go down its concentration gradient, but it can't because it can't cross through the cell membrane. However, it can use a transport protein, and this could be in the form of a channel or a protein that actually just changes conformation and will allow the protein to go to the other side, or excuse me, the item being transported to go through to the other side. So it's still moving down its concentration gradient. That's the important thing. The fundamental difference, facilitated diffusion requires a transport protein, simple diffusion does not. Water, as I mentioned, can actually cross the cell membrane on its own, but it does so relatively slowly because it's polar. So there are particular channels called aquaporins, and they transport water across the cell membrane via facilitated diffusion. And these are necessary in order to transport large quantities of water more quickly. So cells like the kidney that need to move a lot of water around contain a lot of aquaporins. Again, water can get across, but it can get across a lot faster through an aquaporin. Osmosis is a special type of diffusion. It's diffusion of water down its concentration gradient. So let's just review some terminology before we go on to discuss osmosis. First of all, recall that if you have a solution, there are two parts to the solution. The liquid part, which is the solvent, and the solute, which is the substance dissolved in this liquid. Okay, so this is a solution, and within this solution, we have a liquid, which is a solvent, and a solute, which is a substance dissolved in the liquid. And as I mentioned before with diffusion, diffusion is the tendency of a substance to move down its concentration gradient. So if I had a container, and within this container, I had some water, and I just dropped some sugar in the water. This sugar, initially say in a clump, is eventually going to disperse throughout. It's going to move from areas of low, let's say it's glucose concentration, from areas of high glucose concentration, where I dropped it, to areas of lower glucose concentration. And eventually, it's going to end up being evenly dispersed throughout. Now, these glucose molecules might still move around, but they're not going to all clump into one place. Overall, they're going to tend to disperse throughout. So diffusion, again, is the movement of a substance from a region of higher concentration to a region of lower concentration. Now let's apply that to water. Water can do the same thing. Osmosis is the diffusion of water down its concentration gradient. So water is going to have a tendency to move from an area of low water concentration, high water concentration, to an area of low water concentration. So it's going to move down its concentration gradient from a region of higher concentration to a region of lower concentration. Looking at this tube, which is U-shaped, imagine that I had a membrane across here, and it was a semi-permeable membrane. Water can cross this membrane, but the solute that I put in cannot. So imagine that I fill this tube up with water, and it reaches a certain level, and on this side, I put in some sort of solute. It could be salt, it could be sugar, something dissolved in here, and this substance cannot cross the membrane. Water can cross the membrane. What's going to happen is the water is going to move from the area of high water concentration, which is over here on the right. Because on the left, it's lower water concentration because some of this space is being taken up by the solute. So the flow of the water is going to be from this side to the other side. What is going to end up happening, since the solute can't cross, is I'm going to end up with the level of the water rising on the left side as water moves to that side and ending up lower on the right. And this is going to occur until the point at which the concentration of water is, until it equalizes. So let's say I have some solute over here, but a smaller amount. Okay? Then this is going to move the water from this side to the left side until the concentration of water ends up equal on both sides. That's osmosis. And osmosis can also occur in a cell because water can diffuse across the cell membrane. A few terms that you should be familiar with for the tests that relate to osmosis: hypertonic, hypotonic, and isotonic. And when you talk about hyper or hypotonic, you're talking about one solution relative to another solution. So let's say I have two solutions. And the one on the left has a lot of solute in it. This other one, solution 2, only has a much lower solute concentration. If I compare the two, I say that number 1 is hypertonic. So it has a relatively high solute concentration relative to number 2. So hypertonic solution has the higher solute concentration. Hypotonic is the opposite. I say that 2 is hypotonic relative to number 1. So hypotonic indicates a lower solute concentration. Looking over here, this side, number 1, is hypertonic. Side 2 was relatively hypotonic. Now, once the water is done diffusing across and it's equalized the solute concentration on both sides, where you're going to end up is a situation where the two sides are isotonic. This means they have the same solute concentration. At this point, there's going to be no net movement of water. Water may still move across, but there's going to be no net movement. It's going to be overall unchanged. The solute concentration is not going to change overall, although water's may move back and forth. We often talk about hyper, hypotonic, isotonic in biology relative to the cell, relative to the tonicity of the cell. You may hear about experiments, and we'll talk about situations where a cell is placed in a solution. And the solution could be, like in this case, hypotonic. So this solution out here is hypotonic. It is less concentrated, less solute concentration relative to the cell. We could place a cell in a solution that is actually hypertonic. So here is a hypertonic solution relative to the concentration of substances inside the cell. Think about what's going to happen in that situation. If you put a cell in a hypotonic solution, water, as usual, is going to go from a region of higher water concentration to a region of lower water concentration. So with this hypotonic solution, the higher water concentration is outside the cell. That means that water is going to enter the cell, and if the solution is very hypotonic, so much water is going to rush in that eventually the cell will burst or lyse. The cell may lyse. The cell may actually burst because water's rushing in, the volume increases, and eventually the cell just bursts open. Let's look at the situation for a hypertonic solution. In this case, again, water is going to move down its concentration gradient. It's going to move from a region of higher water concentration to lower water concentration, which in this case is going to move outward. Water's going to move from inside the cell to outside the cell. The end result will be that the cell will shrink. If you place a cell in a solution that is isotonic, it has the same concentration of solutes, or we sometimes say the same osmolarity. Osmolarity refers to solute concentration. So if these two solutions have the same osmolarity inside the cell and outside, and it's an isotonic situation, water may leave the cell, water may enter the cell, but there's not going to be any net change. The amount of water that leaves the cell, the volume, is going to be equal to the amount that enters the cell. And the cell size will not change. Cell size or cell volume will not change. So again, three situations. Hypotonic, water's going to rush into the cell may lyse even. It'll get bigger, possibly lyse. Hypertonic solution, outside the cell, water will leave the cell will shrink. An isotonic solution, no net movement of water, the cell size will remain unchanged. Recall that plant cells have cell walls, and one of the functions of the cell wall is to prevent lysis in a hypotonic environment like this. And in fact, in a slightly hypotonic environment, plants can maintain their rigidity. They have turgor from all the water inside the central vacuole pushing against the cell wall of the plant, and it allows the plants to maintain some rigidity so that their stems and leaves maintain their shape. So that's one function of the cell wall. Okay, so this is osmosis. We talked already about passive transport. Passive transport does not require the input of energy. Diffusion, simple diffusion, and facilitated diffusion are both forms of passive transport. Osmosis is the passive transport of water. It's a form of diffusion. Active transport requires the input of energy in order to move a substance against its concentration gradient. Recall in diffusion, a substance is moving down its concentration gradient. It's moving from higher to lower concentrations, just like it wants to. So first looking at regular active transport, not yet at cotransport. Consider a substance such as sodium. And there is a relatively high concentration of sodium outside the cell. There's a relatively low concentration of sodium inside the cell. Potassium is the opposite situation. There's a relatively high concentration of potassium within the cell and a relatively low concentration outside the cell. So looking at which direction things are going to want to move in. Sodium is going to want to go into the cell. Potassium is going to want to leave the cell. It's going to want to go down its concentration gradient, and so is the sodium. However, in order to maintain the correct concentrations of these ions in the cell doesn't just allow them to move in the directions they want to go. Instead, there's what's called a sodium-potassium pump that maintains the correct balance of sodium and potassium. And this is done through active transport. Recall first that since sodium and potassium are ions, they can't just cross the cell membrane on their own because of their charge. Also, because I want to move them against their concentration gradient, energy is going to be required. So ATP will be required. The sodium-potassium pump pumps sodium out of the cell. So the sodium is going to be pumped out of the cell at the same time that potassium is being pumped against its concentration gradient, and it's being pumped into the cell. And for each ATP that is used, 3 sodium ions are pumped out for every 2 potassium ions that are pumped in. So 3 sodium out for every 2 potassium in. Again, this is going to require ATP. So ATP is going to have to be hydrolyzed to ADP to provide the energy for this process of pumping sodium out of the cell against its concentration gradient and pumping potassium into the cell against its concentration gradient. There's another consequence of this. So if 3 sodium are being pumped out for every 2 that are being pumped in, there's a net loss. This means that there is a loss of 1 positive charge for each cycle. So for each ATP used or each turn of this pump, there's a loss of 1 positive charge because 3 positive charges, 3 sodiums are going out, only 2 are coming in, net loss of 1 plus out. The result is going to be that the cell has a relatively negative charge compared to the outside. That charge difference is called membrane potential. And we'll talk about this in more detail when we talk about the neurological system, the nervous system. But different cell types have different membrane potentials. For example, a typical neuron has a membrane potential of approximately minus 70 millivolts. And that is maintained by the sodium-potassium pump in animal cells. In plant cells, fungi, bacteria, the membrane potential is actually maintained by a hydrogen pump. So the hydrogen pump maintains the membrane potential, this chemical gradient, electrical chemical gradient. So because there's a gradient of sodium, potassium, or hydrogen, as well as an electrical gradient, this difference in potential. So the hydrogen pump does the work in plants, animals, and fungi, whereas it's the sodium-potassium pump that mainly maintains that gradient in animal cells. Okay, so this is called primary active transport, because in this case, the ATP is being used directly by the transport protein. There's a second type of active transport called cotransport. So let's look over here on the right at this purple protein that's going to be the cotransport protein. Cotransport also uses energy, but it uses it in a slightly different way. Cotransport uses the energy of a substance moving down its concentration gradient to move another substance that's moving against its concentration gradient. So what it does is it harnesses the energy from one substance that's going the direction that it wants to go, and it uses that to push a substance going against its concentration gradient. A typical example would be glucose. So glucose is involved in cotransport, is often cotransported. The sodium glucose cotransport occurs in the kidney. So let's say that I've got over on this side this high concentration of sodium. And sodium really wants to go into the cell where there's a low concentration of sodium. This co-transport protein, so this is going to be the sodium glucose co-transport protein. The sodium wants to go down its concentration gradient. So as it's moving down, it's releasing energy that's harnessed to move glucose. Let's say that the glucose has a high concentration in the cell, for example, and a lower concentration outside the cell. Then glucose does not want to enter the cell, so it has to move via active transport. So if you look at this, the sodium and the glucose are moving in the same direction, but the sodium is moving down its concentration gradient, whereas the glucose is moving against its concentration gradient. So the glucose requires energy to move in that direction, whereas the sodium does not. And actually, if there was a high concentration of glucose out here and the glucose needed to move in the other direction, the sodium still could move down its concentration gradient and provide energy to push the glucose the other way. So glucose and sodium, or whatever two things are being transported, in some systems they're physically being transported in the same direction, in others they're not. All that matters is that one is going down its concentration gradient and the other is going against its concentration gradient. So I said that active transport requires energy, and here I'm talking about the release of energy. So where is the energy used? Where does this energy come from? Well, it comes from this earlier step. In order to maintain the gradient of the sodium in this case, if I just let this go, eventually more and more sodium would come in until I had an equal concentration of sodium inside and out of the cell, and there would be no net movement of sodium to harness in order to transport the glucose. So for this pump to continue to work, or this co-transport to continue to work, the cell has to maintain the sodium gradient. The energy for co-transport is used in maintaining the gradient of the substance moving down its concentration gradient. So it's in an earlier step. So there's two types of active transport. Primary active transport, in which the energy is used directly by the transport protein, and secondary active transport, in which the energy is used at an earlier step, which is to maintain a concentration gradient for a substance that will eventually move down its concentration gradient. Okay, so these are two types of active transport, and we also talked about a couple types of passive transport. So active and passive transport are methods through which substances, molecules can move into the cell or out of the cell. Now, if a cell needs to uptake larger substances, such as proteins, or release larger substances, such as proteins, it usually uses endocytosis and exocytosis. These both require energy. So they require energy, usually in the form of ATP. First, looking at endocytosis, which is shown here. So here's the inside of the cell. This is the cytoplasm. Out here is the extracellular space. And we see this particle, and it's moving towards the cell. The cell membrane invaginates and actually pinches off, as shown here, to form a vesicle. And then that vesicle ends up in the intracellular space. So this is called endocytosis. And the opposite process can occur. So if I actually went from right to left, if I started out with the substance in the cell, and then so if I had a substance in the cell, and it was packaged in a vesicle, that vesicle could actually fuse with the cell membrane, and then be extruded and released outside the cell. And that is exocytosis. For example, exocytosis could be used to release a protein that was made on the rough endoplasmic reticulum. That protein would then be processed in the Golgi apparatus, packaged in a vesicle, and then it could be released from the cell, or sometimes this vesicle will fuse with the cell and the protein will just end up on the cell membrane. So this is how cell membrane proteins end up integrated into the membrane, or you could have a substance that's actually being released completely. Neurotransmitters are released. So example, neurotransmitters are released through exocytosis. Also, cell membrane proteins. Endocytosis can be used to uptake substances such as invaders. So cells of the immune system, leukocytes, white blood cells, may ingest invaders, pathogens such as bacteria. And then the vesicle will fuse with a lysosome, which contains enzymes that can digest this invader. There's a couple types of endocytosis that you should be familiar with. One is pinocytosis. This is sometimes called cellular drinking because it is the process through which the cell ingests liquids. So if the cell pinches off a vesicle like this and it contains a liquid, that's called pinocytosis. Phagocytosis is sometimes called cellular eating. This is when solid particles are ingested by the cell through endocytosis. And what I just described here with a cell ingesting bacteria, a leukocyte, a white blood cell ingesting bacteria, that would be a type of phagocytosis. There's another type of endocytosis known as receptor-mediated endocytosis. And what this allows is for the cell to take up specific molecules. So the receptors shown here have specificity, the same way that an enzyme has specificity for its substrate. Here we have a substance that's going to bind, and this substance that binds is going to be, once it's bound, called the ligand. So you have a receptor and the ligand, and these two are specific. They are shaped and they have a specificity to one another, so that instead of just ingesting non-specific molecules, this allows certain molecules, such as a good example is LDL, cholesterol, to be ingested. So here you see the receptor binding to a ligand, and let's use LDL as an example. And these are LDL receptors, and LDL, the cell, again, the plasma membrane invaginates. It's going to pinch off and form a vesicle in the last step. And this type of vesicle has the name coated vesicle because it is coated with its receptors, bound to its ligand. And then these can be released within the cell, and the receptor, and then this vesicle can actually go back and the receptors can be recycled, or they can make their way back out to the cell membrane in a vesicle, be reused, ingest more ligand. So again, LDL is a form of cholesterol found in the body. That it stands for low density lipoprotein. And there's actually a disease called familial hypercholesterolemia, which is caused by a defect in the LDL receptors. So some people have a genetic defect in their LDL receptors, and the result is they don't have enough receptors or enough functional receptors, so they don't uptake LDL. If there's too much cholesterol left floating around because the cells aren't taking the LDL up, the result can be heart disease. So early onset of heart disease due to high cholesterol, and that's a result of a defect in these receptors. Let's look at some examples to sum up what we've discussed today. Example 1, list 3 characteristics of substances that are permeable to the cell membrane. So what type of substance would be most permeable to the cell membrane? Well, first of all, something that is small. Small substances are most easily ingested. So size. Second would be polarity. Nonpolar molecules, such as hydrocarbons, are much more able to cross the cell membrane because of the hydrophobic interior of the phospholipid bilayer. The third characteristic is charge. So actually an uncharged molecule is going to cross much more easily than an ion. Again, this is because of the hydrophobic interior of the phospholipid membrane. So molecules that are permeable to the cell membrane are likely to be small, they're going to be nonpolar, and they're going to be uncharged. Oxygen, CO2, water can cross because even though it's polar, it is quite small. Hydrocarbons, these are all more readily permeable to the cell membrane. Example 2, an animal cell is placed in pure water. What would be the outcome and why? Let's sketch this out. So pure water. Now I put an animal cell in it. And the animal cell is going to have all kinds of solute substances in the cytoplasm, whereas the pure water is not. Therefore, what you've done is to place the cell in a hypotonic environment. This is hypotonic relative to the solute concentration, the osmolarity within the cell. Remember that water wants to go from high concentration of water to a relatively low area of low concentration of water. So out here, there's a very high concentration of water because there's relatively few solutes, whereas inside, there's a lot of solutes. So water is going to rush into the cell. The cell is going to become larger and larger, and it's eventually going to burst or lyse. So cell takes up water. So water is going to cross the cell membrane. So via osmosis, water is going to cross via diffusion, via osmosis. Cell will take up water and eventually lyse or burst. Now let's say we repeat this procedure in a plant cell. What would possibly happen? And why would this outcome be different? Again, I have a hypotonic environment, but now I have a plant cell. And recall that an important difference between a plant and an animal cell is the presence of the cell wall. Therefore, water is going to rush in, and the cell will increase in size, but it won't lyse. Plant cell will not lyse due to the cell wall. The cell wall provides the strength and the structure so that this cell will not just keep on absorbing water to the point that it actually lyses. Example 3. Why isn't energy required for passive transport? Recall that in passive transport, substances are moving down their concentration gradient. This is the direction that a substance wants to move in. So if there's, for example, a high concentration of sodium, sodium is going to want to move to an area of low concentration of sodium. Sodium will want to move in this direction. That is not going to require the input of energy because it wants to move in that direction. Whereas if the sodium were to move in the opposite direction, energy input would be required. What is the source of energy used to drive a molecule against its concentration gradient in cotransport? So how are molecules, substances moved against their concentration gradients in cotransport? Well, energy from another substance moving down its concentration gradient is harnessed to move a second substance against its concentration gradient. Remember that it requires energy to maintain the concentration gradient of the first substance. Otherwise, that concentration gradient would cease to exist. But the immediate source of the energy during co-transport is the release of energy from a substance moving down its concentration gradient. For example, sodium moving down its concentration gradient provides the energy to move glucose against its concentration gradient in sodium-glucose co-transport. What is the difference between simple diffusion and facilitated diffusion? Facilitated diffusion requires a transport protein. Facilitated diffusion is used to transport substances that cannot cross the cell membrane on their own. Maybe they're too large, or they're polar, or they're charged. How are they the same? Well, both do not require energy. In both facilitated and simple diffusion, substances are moving down their concentration gradients and they do not require the input of energy. The difference is that facilitated diffusion requires a transport protein. Example 4, match each of the following terms with its definition. And we have exocytosis, pinocytosis, receptor-mediated endocytosis, phagocytosis, and regular endocytosis. Looking at the possible definitions. Molecules bind to specific receptors on the cell membrane and then enter the cells via phagocytosis. So if the molecules are binding to specific receptors and endocytosis is occurring, that would be receptor-mediated endocytosis. The example I gave was with LDL. They bind to LDL receptors and are ingested into the cell via endocytosis. B, molecules contained in vesicles leave the cell by fusion of the vesicle with the cell membrane. So if a molecule is leaving, or substance is leaving the cell in a vesicle, that's considered exocytosis. So it's exiting exo. If it's entering the cell, it's endocytosis. Therefore, exocytosis, number 1 up here, is B. The uptake of fluids by the cell through a form of endocytosis. Recall that there are a couple types of endocytosis. One is considered the cell drinking, sometimes cell ingesting fluid by endocytosis, and that is pinocytosis. D, the uptake of particles of the cell through endocytosis. Again, a particular form of endocytosis, but this time instead of fluids, we're talking about particles, is sometimes called cellular eating, and that is phagocytosis. Such as when a macrophage or leukocyte ingests bacteria through formation of a vesicle from the cell membrane. Finally, molecules enter the cell by formation of vesicles derived from the cell membrane. So just the general term is endocytosis. Okay, so we have all of those taken care of, and that concludes this lesson on educator.com. Thanks for visiting.